Hey everyone, my name is David Eberts, and we're going to look into the main components of the ECM and the few things that you need to know about each one of them. So the four main components of the normal extracellular matrix is collagen, proteoglycans, lamina, and fibronectins. So let's start at the top with collagen. As we know, collagen is a trimer of protein. Is a trimer of glycoproteins. And glyco just means it's a sugarous protein. Don't confuse that with proteoglycans. This is a specific type of protein that has sugar in it, and we'll discuss it later. So collagen is a trimer of glycoproteins, and so it looks almost like DNA, but triple-stranded instead of double-stranded of similar or dissimilar collagen. And what they do to the cell is they give strength and resistance to pulling forces. So they have a wide array of uses. Because of this trimer, there's so many different conformations. We have many functions that they're attributed to, functions. But the main one for in the extracellular matrix and in the basement membrane are strength and resistance to pulling forces. So the ECM is nice and strong, has a nice stability and foundation. To add on to that then, we have proteoglycans, which we looked into as dealing as a core protein at the center. So now we're looking at proteoglycans. There's a core protein at the center, and on the ends of it, or on the sides of it, we have these sugars, and that's the basic formation of it. However, there's a bigger macro volume of it when it's attached to a non-sulfur containing hyaluronic acid, so HA. So this is going to get even bigger. So hyaluronic acid, that each one of these can come on and attach to the side and become a giant glycoprotein, or a giant proteoglycan. And just like on each one of those, there's the hairs. So you get the picture of what that is. Now what it adds to the extracellular matrix has to deal with this component right here, and this is called a GAG. And it's a glycoaminoglycan is what that stands for. You don't need to worry about that too much. But basically, all these sugars that are attached here, we have sulfur or carboxyl sugar. So there's sugars on all these little red strands that have sulfur or carboxyl on it, which are very negatively charged. And so all these negatively charges, which I'll draw just in green, just on those ones right here, but it's everywhere on these red strands. What it'll do is it'll cause cations to come over. So cations to flow towards wherever these GAGs are. So all these GAGs will attract cations all over the proteoglycan. And whenever cations are brought over and involved, you can bet your bottom dollar that H2O is coming with it. So the presence of the glycoaminoglycans, or GAG, and the fact that they're negatively charged sulfur or carboxyl regions will bring over water, and what this will do is you'll essentially have water all over this huge proteoglycan. So let's see, that was there. This is a proteoglycan. And because of this, you're going to have water all throughout here. I'm not going to draw it in all the way, but so it brings water in because of its negative charge. And so what that does is that proteoglycans attribute to the ECM a gel-like structure, a gel-like almost packaging. And so you can imagine it's kind of like a foamy, liquidy substance. It makes it really safe for the cells to move and get compressed on without being damaged. So gel-like support. And so this complements really well with collagen, which adds strength and resists pulling. Together, these two things make a really nice extracellular matrix where it's strong, it can be pulled left and right and be resistant to that and not be pulled apart because collagen's holding everything together. And then proteoglycans give it a nice cushioning so that everything doesn't get squished or pushed around too much. So it's a really nice complement and gives the extracellular matrix some real integrity. Let's see, so that's proteoglycans. Isn't it always awkward when professors are erasing boards? I think so. I don't know, it's just like, what to talk about, what to do, blah, 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 blah. Alright, there we're done. So, collagen, check. Proteoglycans, check. Lamina. So, lamina are a tripolypeptide as well, and it looks like a cross. So you have one large protein with two smaller attached proteins, and they're all attached via disulfide bonds. Disulfide bonds. That's not too important, don't worry about it, but it's a trimer protein. And what it can do is it can interact really well with collagen. So collagen, as we said earlier, is a trimer. And if we were to look at that a little larger, we can imagine these huge strands of collagen that just form this great lattice network that the cells can sit on in the extracellular matrix. And so, let's change the color of this lamina real quick to orange. And so what can happen when we have this collagen that's making these huge things, adding a resistant force and everything, adding strength, is lamina will come in and can bind in between it and bind with it, creating all these cross bonds and double bonds and triple whatever bonds, doesn't matter. But it could come in and continue to make a lattice structure. And essentially, this now complements collagen to complete a net almost that the cells can sit on top of, and makes it really safe for the cells and really secure, and adds to the integrity of the basement membrane. And so it's hard for cells won't fall through it or out of it. And it's also important in cancer because cancer cells can't penetrate it too easily. It's just adding strength and integrity to the basement membrane with collagen. So lamina contributes to a lattice structure with collagen. Yes. So they bind to cells and cells sit on top of there and they're happy. Great. Now let's move on to the final one, fibronectin, which I think is a really cool thing. So we're done with lamina. Now fibronectin. Fibronectin is kind of a identical, repeated protein that has multiple regions for different interactions. So here's one region, we'll call this, which we'll talk about, the cell binding domain. And then you may have just other different regions that do different things, and everything's kind of a mirrored image on this. So it has this two structured formation that's brought together by sulfur bonds over here. Not important once again, but we want to focus in on the cell binding domain area because that's where the ECM is so important. Inside this area of the cell binding domain, we have the RGD loops. So color this black right here. Everything's identical and mirrored on both sides. We have an RGD loop. And what that stands for is arginine glycine aspartic acid. Those are just the single abbreviations for the amino acids in this region. Don't worry about it, but remember RGD. So the RGD loop is the actual part that can bind cells. And so when a cell wants to connect with the ECM fibronectin, it'll bind onto this specific part. And we find out later that other structures such as these will have an RG loop that can bind to things. I think lamina has one as well. But anyway, this is also really important. So what fibronectin does, once you have cell binding, allows movement of cells. Oops. Movement. And then the other important thing is branching, which we spent the whole day on. And so I'm going to draw it now a little more macroly, just so you can get the concept down. Now imagine that we have cells, just a cell like this, and it wants to move this way. Well, one way to do it is to interact with its extracellular matrix. And so say we now have collagen down here, fibronectin, which I'll draw like that, fibronectin. I'm going to draw it like this. You'll have it all different places in the ECM. And what the cell can do then is it could reach out with certain proteins, integrin, which can be seen in another video, can reach out and interact onto that RGD loop, RGD. And then it could reach over and grab onto another one, and that'll move this. And then it could reach out and grab onto another one and pull it and move it. And essentially you just get this reaching out, pulling, and movement. And that may not exactly be the exact mechanism, but just know that these are seen in the highways of the germ cells that we looked at, and when things are in development. They like to move along areas rich in fibronectin. So it helps with movement, as well as, oh, and on that note, I apologize, lamina is also important in movement. These two, there's a portion in our book that says these both, when cells were moving, they were found in lamina and fibronectin-rich environments. So both of these can do movement, as well as how lamina added strength to the basement membrane. Then we saw in our book that branching can occur. We saw in our example in class that it could lead to branching. So you have two cells that are adhered nice to each other. One thing involved in it is fibronectin. So we have fibronectin coming in here like this. And so I opened it up a little bit, and as these cells try to branch away from each other, let me keep the colors consistent. You'll see that fibronectin is all in throughout here. And it may be pulled out a little bit and extended. But the important thing to note is that fibronectin is found within branching, and as always, if it's interacting with the cells, it's going to be at an RGD loop. So interaction at RGD loop. And as you saw, a really versatile thing about fibronectin is that since it has that repeated formation, if it gets pulled open and essentially gets opened or stretched, it opens up more binding domains. So maybe a binding domain that was in here is now exposed and can do even more binding to other different things and functions. But yeah, so movement, branching, and we also see it later in talking about platelet aggregation, and basically it holds all the platelets together, and there'll probably be a video about that. So those are the main things you need to know about collagen proteins, lamina, and fibronectin, which are the main components of the ECM, and how they interact with cells or give strength or versatility to cells, whether it be movement, resistant force, or cushioning by proteoglycans. All right, I hope that helped.